Hello everyone, welcome back to another video from Shomu's Biology. In this microbiology lecture, we are going to talk about the Streptococcus pneumoniae characteristics. Everything related to the Streptococcus pneumoniae that you need to know to get a basic idea about the Streptococcus pneumoniae bacteria with all the details. So let's start with the first thing, that is the gram nature of Streptococcus pneumoniae, and we know for simple that the Streptococcus pneumoniae are gram positive in nature. The second important point is the disease caused by Streptococcus pneumoniae, and it is a pneumococcal infections that including pneumonia primarily, and otitis media can also be caused, sinusitis can also be caused, and meningitis can also be caused by Streptococcus pneumoniae bacteria. The next one is the virulence factor. What are the virulence factors for Streptococcus pneumoniae? And the list are a capsule. Capsule is a virulent factor for Streptococcus pneumoniae that impairs phagocytosis. That is a major virulence factor. The second one is a pneumolysine, cytotoxin that damages the host cells. And the third one is autolysine, facilitates the release of cell wall. What autolysis does is facilitates the release of cell wall components, promoting the inflammation and inflammatory response in the host body. Next is the incubation period for the pneumonia bacteria. The incubation period variable, but typically it ranges from 1 day to 3 days. Okay, for respiratory infections. Okay. The next one is the antibiotic susceptibility. What are the antibiotic this Streptococcus pneumoniae is susceptible to? There are a list of antibiotics it is susceptible to, like penicillin, it is susceptible to cephalosporins, macrolides are the types of antibiotic it is susceptible to. Now, antibiotic resistance property of the Streptococcus pneumoniae and increasing resistance of antibiotic against some antibiotics like penicillin, macrolides are visible. Okay, that is a matter of concern. What are the culture methods for Streptococcus pneumoniae? The cultured methods that we use, it can grow on standard culture media. Now, what are the standard? Like blood agar is a standardized culture media where Streptococcus pneumoniae can be cultured. It forms alpha hemolytic colonies. That is a special type of colony which will help us to detect the presence of Streptococcus pneumoniae by looking at the plates, agar plates. Detection method for Streptococcus pneumoniae. Here are 4 detection methods. The detection method number 1 is the culture methods. The simple blood agar culture. We can isolate Streptococcus pneumoniae from clinical samples using the blood agar culture method. The second method that we use is the optogene sensitivity test. What is optogene sensitivity test? It differentiates the Streptococcus pneumoniae from other alpha hemolytic streptococci, because Streptococcus pneumoniae is not only the bacteria that can break down the red blood cells in the agar. There are other bacteria who can do that, but this can differentiate from the other alpha hemolytic streptococci as well. The third important detection method that we can use are Quellung reaction. What is Quellung reaction? It detects the presence of the pneumococcal capsule in that sample. So in the sample, if there is a particular specific pneumococcal capsule found, it can be detected with Quellung reaction. And the fourth detection method that we can use here is a molecular test. PCR can be utilized to detect the presence of the specific nucleic acid structure provided by the Streptococcus pneumoniae. So these are all the 4 different detection methods that we have. So this in a sense is a basic overview of the Streptococcus pneumoniae characteristics and the details about Streptococcus pneumoniae microbiology. If you like this video, please hit the like button, share this video with your friends, subscribe to this channel to get more videos like that in future. Thank you. Bye.